Okay, this is Chapter 3, Part 2. We talked a little bit last time about issues of validity, reliability, and got into experimental methods a little bit. We start up with the PowerPoint slide here, listing major types of research design. We already talked about experimental. We'll talk about that in more detail here in just a moment. Cross-sectional designs, longitudinal designs, case study designs, and comparative designs. Now, again, none of these is perfect. They all have their strengths and weaknesses. And just a reminder that as you take notes or as you get yourselves prepared for any quizzes, it's always good to know strengths and weaknesses of these methods, because no method is perfect, and what's the best method to use depends on your situation, depends on what research question you ask. So it's important. In this course, we want to introduce you to all these different types of methods and the pros and cons, so that if you ever need to do a study for a different class, that you'll have some sense of what's the best way, what the best method to use for your situation. And also, if you're reading studies done by other people, which you do all the time in various classes, textbooks are filled with studies, is you should be able to develop some critical skills to be able to tell whether they did a good job and chose the right kind of methods for their situation, for the types of questions that they were asking. So again, let's get into the different types, starting out with experimental. In this chapter, I want to spend the most time talking about experimental methods, because we don't have a chapter devoted entirely to the experimental methods like we really honestly should. And the reason for that is that disciplines like sociology and social work don't use experimental methods very much. That's mostly psychology that does that. And so you get more of an emphasis on experiments if you take a research methods class from psychology. But it's still a very important method, and so we need to cover it in a fair amount of detail. The experimental method is really considered to be sort of the gold standard of studies. And I'll say right up front that a real strong positive, a strength of experimental methods are that it's typically the method that's the best way to tell what is causing what. And so, of course, the classic example of where you see experiments done all the time is in medical research. So, say, for example, if we're trying to figure out if a new therapeutic, a new medicine is going to help reduce the symptoms of COVID-19, something's going on right now, you need to, you're going to use experimental method. You're going to, and so the way that you do the core, I talked about Bandura before. Let's use another example. Stick with this medical kind of example. What you do in a classic experiment is you get a large sample of people, hopefully sort of a sample of people that are representative of your entire population. So if you are trying to do a test to learn if some drug was effective, you'd want to have a nice random sample of individuals, a large sample, and then you would want to randomly assign those individuals to one of two groups. The two groups I mentioned before for experiments, you have an experimental group and a control group. And the only difference between those two groups is that the experimental group is the group that gets the medicine, that gets this. Sometimes the term is used that they get the treatment, they get the intervention. It even sometimes gets called manipulation, which doesn't sound so good, but it's the thing that the researcher's giving to the group or doing to the group to see if there's a response, to see if it has the effect, to see if the medicine reduces the COVID-19 symptoms. And so you want to have those two groups the experimental group and the control group to be as equivalent as possible, meaning the same. The two groups look the same. They both have similar numbers of males and females, young people, old people, older people. You know, the mix of people should be the same for the two groups. If it's not, then you got a problem, because if you have all men in one group and all women in the other group, and the men turn out differently than the women, well, maybe it's because they're men and not because of the medicine that you gave them, right? So you want to equalize the two groups, and one of the main ways that you equalize the two groups is this random assignment that the notes say here, the PowerPoint slides say. You want to randomly assign people to the two groups. So you want to do something like have random numbers in a hat. People pull a number out of a hat, and you assign people that way. Or, you know, something like that that doesn't favor people, but just randomly, through chance, puts them in one group or the other. You don't want to favor people. It's all supposed to be random. That's important because at least if you have a big sample, say 500 people, and you're going to put 250 in one group and 250 in the other, if you do that randomly, those two groups should look very similar to each other. Not perfect maybe, but they should be fairly close, because you're not hand-picking people. It's just a random assignment. So that's important. That's key for the experimental design. Now it talks about pre-testing and post-testing. And all that means is, well, just like we talked about measurement before, you have to measure carefully and validly whatever it is that you're studying. So let's say that a lot of this big sample of people, some of them have COVID. Well, let's say all of them have COVID symptoms. They've all been diagnosed with COVID-19. They've got fever, and they got chest problems, respiratory coughing, and pressure in the lungs, and all these other sorts of symptoms. And so you need to measure all of those before you give them the medicine to find out if they improve. If you don't measure pre-test, then how do you know if they got better? So you measure them before, you measure them after. That's the pre-test and the post-test. So make sure that you understand what they mean when they talk about pre-testing and post-testing. You have to measure before and after to know if there's improvement or not. And then you have this term of independent variable, and what that means is that they use this term manipulation. The researcher is manipulating the independent variable, which is just a fancy way of saying that the researcher is giving different conditions for the two groups. The experimental group gets the medicine, the real thing. The control group gets a pill too, because you want to equalize everything that you do in one group, you want to do the other group. That's the equivalence thing I was talking about. You want to treat the two groups the same, except for one thing, the medicine. So you go ahead and you give the control group a sugar pill that's called a placebo, a fake pill. And the reason you do that is if there was any benefit psychologically from taking medicine. There's some research that indicates that if you believe you're going to get better, you believe you're going to improve, that you tend to improve. And so if you're taking medicine, you might believe it's going to make you better, and it does improve your symptoms, but it was done through your belief, your psychology, not because of the medicine, because there's no medicine in the pill. It's just a sugar pill. It's a fake pill, right? So that's what they mean by placebo. So the researcher is manipulating the situation. He's giving different conditions to the two groups. One group gets the medicine, the other one doesn't. So the variable here is whether you get the medicine or not. The independent variable. One group gets it, the other one doesn't. So that's what they mean by variable, because it varies from one group to the other. And independent means that you think it has an influence on the people's health, on their symptoms. So that's what I mean by independent. It's having an influence on something. The dependent variable, which is whether the people's symptoms get better or not, whether their symptoms improve. That varies from person to person. It could vary from the experimental group might get better and the control group might not. So independent variable, dependent variable. Independent is having an influence, dependent variable being influenced upon, right? So the drug reduces the symptoms, independent variable, dependent variable. Okay, so you go ahead and you give that medicine to the one group, and you watch them over a period of time, and then you measure their symptoms and see if the experimental group had improved symptoms compared to the control group. And you collect data. That's this last point here, computation analysis of group differences. Did the experimental group that got the pill, the actual medicine, did they improve? Did their symptoms get better than the symptoms of the average person in the control group, the people that didn't get the medicine?